in terms of the Q&A, right, that I post at the end of the classes, there's been a lot of people that have been saying, man, just linking all these concepts together, right? And the context, of course, for that is glycolysis and gluconeogenesis and glycogen breakdown and glycogen synthesis and all those things, right? Different tissues and different conditions and the regulation of those things. Okay, so I kind of want to spend some time drawing out a big map and trying to help people visualize and conceptualize these things and their relationship to one another in the relationship of some of the bigger concepts we talked about, okay? And like I said, I think there's quite a bit of people. In fact, in this last Q&A that I had 10 minutes ago, there was 12 to 15 people that said linking these concepts together is just really hard. So I'm going to do that, okay, to kind of start off. Now, if you have questions, right, specific questions that are not just linking them together, by all means, I want you to be able to ask those today and get those questions answered. But let's start off with that linking them together, that kind of a map, okay? So I'm going to turn off my video. Sorry, I turned off my mic. I'm going to turn off my video, and then I'm going to share my screen with you, okay? Let's see here. Okay, screen one. There we go. Okay, so where to start something like this? As I was walking back from class, I thought, okay, how can we do this? And so let's try and conceptualize this. We're going to need a lot of room here. So let's try and conceptualize this first of all, because we're talking about carbohydrate metabolism. We'll just think here real quickly about glucose. Okay, obviously. And I'm going to put glucose on the outside of the cell. Okay, so do this. And then we'll have of course, a membrane transporter to get glucose inside the cell. And we'll move it in, right? So of course, glucose is going to come into the cell. Okay, and I'm just going to use the abbreviation GLC. Okay, now, there are lots of different things that can happen in a cell to glucose as it moves in, or that can happen in a cell to move glucose out. So let's just go real quickly think about this to begin with, okay? And that is that glucose could become glucose 6-phosphate, okay? Or it could be broken down to glucose. Okay, so I'm going to put some of these other steps here. And I'm not going to write all of glycolysis out, but I am going to do a little bit more detail in this. Okay, so then this could become fructose 6-phosphate. Okay, reversibly, and then this could become fructose 1,6-bisphosphate. Or it could be broken down. Okay. And then I'm going to put kind of a big double-headed arrow till finally we get to PEP at the end of the pathway, and then that can be broken down to pyruvate. Okay, and that can be converted to oxaloacetate back to PEP. Okay, right. So what I'm showing here would be the two opposing pathways that we've talked about. Okay, and when people say, well, linking all these concepts together, the first thing I think to myself is, well, you got to start with the pathways. You got to understand how pathways are related to one another before we can really think about how they're regulated. Okay, so what I just drew, the blue arrows headed upwards would be the irreversible steps of gluconeogenesis. The black ones headed down, single-headed arrows would be glycolysis, right? So that's essentially one of these pathways, okay, or two of these pathways, gluconeogenesis and glycolysis. Okay, all right. Now, at the same time, right? I guess maybe we should say this because it's important. We should also put ATP production here. Okay, and really it's 2, same thing here, 2 ATP. Okay, and that has a really important bearing in muscles. Okay, now the other thing here is this. Okay, and that would be the production of essentially glycogen. Okay, that wasn't a very good arrow. Let me try that again. So I want to link these. Actually, let me use a different color. That way we kind of keep these segregated in our minds here. And yeah, I guess a different color would be fine. Okay, so kind of get this away from this. And we'll put G1P here, okay? And then what we'll do here is, let me see, what's the best way to go ahead and draw this out? G1P can be used to make, okay, glycogen okay, and I'm going to just put N plus 1, right? And then I'm going to put, coming into this reaction, glycogen, N, okay? So we're adding one of the glucose molecules, okay? The enzyme that's going to do that is glycogen phosphorylase, right? At the other end of this, I'll go ahead and use blue for this. We can break this off, okay? And this would be glycogen N, okay? So I think I misspoke. I meant to say that this would be glycogen synthase up here, okay, in the purple, and then in the blue, the glycogen phosphorylase. We're skipping some things here, clearly. We're skipping branching steps. We're skipping UDP, UDP glucose phosphorylase, pyrophosphorylase. So there's some things that we're missing here, okay, but this is, I think, good enough to try and help link some of these concepts together, okay? Okay. Now, when you have this kind of thing visualized in your mind, you can start to better understand how we link all these concepts of regulation together. Okay. Now, before I go on, are there any questions about that? Because if you don't have this down, it's really hard. Okay, doesn't seem like it. Okay, great. Okay, so let me do this. I'm going to copy all this because it would be great to have that for a different duplicate. There you go, we'll move it down there. Okay. Oh, I didn't get all of it. That's okay. We got the major portions of it. Okay, so we'll use that. Wait, what happened to it? Crud. Delete. Let me try it again. Delete. Delete. This will pay dividends later on. Trust me. Yeah, there we go. Copy all that again. Okay, because what we want to try and do this in is both muscle and liver. Okay, there we go. Right. All right, so let's go back up here now, okay? So we've got this whole thing now, and let's then think now about the liver, All right, so this is the liver cell that we're drawing this in, okay? And let's think about this, that you're headed into a fasted state, okay? In other words, glucose concentrations, and maybe it's even best to just not have this arrow here. Glucose concentrations on the outside of the cell are going like this, okay? That's right, a condition that happens, of course, if we haven't eaten. Okay, so that means that the liver needs to do this. It needs to take glucose and move it out, which means that it needs to activate, and let me put stars by this, it needs to activate these processes, okay, where I just put those green stars, and then it needs to inhibit these processes, okay, that are going to essentially use glucose, okay, either through glycolysis, meaning that the liver cell would start to bring it in, and, okay, make glycogen from it, right? So you don't want to do those things, okay? The question then, and this is what, we're kind of working our way through, is, well, how is it going to do that? Okay, how is it going to activate glycolysis or gluconeogenesis and activate glycogen breakdown? Well, we have here, okay, let's see here, let's put another receptor over here, or another cell something, okay, and let's call this the glucagon receptor. Put that title out here. Now I'm going to naturally skip over some details in some of this, okay? But glucagon receptor, right, when this starts to happen, glucose goes down, that will release, glucagon will be released from the pancreas. And glucagon is a peptide hormone, okay, released from the alpha cells of the pancreas, will bind here, and then that's going to lead to, right, I mean, I'm not going to draw all this out, but that's going to lead to the G protein changing conformations in the alpha subunit. It will exchange GDP for GTP. That alpha subunit will let go of the G protein. It will then traverse the membrane. It will find adenylyl cyclase, which will make cyclic AMP. Okay, so kind of downstream of this would be cyclic AMP will go up, and then this would then activate pKa. Okay, and we'll put a little green circle in it. Activated pKa. Now, activated pKa is going to do lots of stuff. Okay, and so, but we want to try and kind of draw this in this big diagram to understand how all these concepts are related to each other. Okay, so pKa now is going to first of all do this. Okay, it is going to then, I'll use a green arrow to indicate that it's going to do this. It's going to take that bifunctional enzyme PFK2, FBPase2, and it's going to phosphorylate it. It's going to phosphorylate it on the kinase domain. Okay, and so now we'll have this phosphate on it. That's what pKa does. Okay, sunbeams, we get a sunbeam, we get to draw sunbeams, right? So sunbeams coming out of this now, right? That is going to then take this molecule I'll use this, fructose 2,6-bisphosphate, break it down, and break it down to fructose 6-phosphate, okay? Now you say, yeah, that's right. That was like last week's worksheet or something along those lines, and that's exactly right, okay? This molecule, though, is going to do this, okay? If we look at it this way, how can I draw this out? Let's use the same colors. But fructose 2,6-bisphosphate, we put it here, it inhibits this enzyme and it activates this enzyme. Okay? Oh, no, I put it in the wrong place. That's hexokinase. Shoot. Sorry, everyone. Don't draw that. It's easy for me to erase. Maybe not so easy for you. Sorry. I'll put it down here. Fructose 2,6-bisphosphate. Okay, like I said, it will inhibit this enzyme and it will activate this enzyme. Okay, but we're losing it, right? So this is going essentially down now because we're breaking it down because of everything that's happening out here, right? Which means that if we're losing an inhibitor, we're activating our gluconeogenic pathway, okay? Now, there's other things that would happen, of course, okay? But this is certainly one of them, okay? Right. Now, pKa is going to also do this, and this is where it's going to be like super fun, okay? It is going to also do this to phosphorylase kinase. So I'm going to use some abbreviations because I have to. It just gets too much to write all this down. Okay, so I'm going to use phosphorylase kinase as my enzyme here. Okay, and this is going to, my green arrow is not quite right. Let me draw that. Let me draw that again. Okay, it's going to pKa because it's a kinase, phosphorylase kinase. Okay, and when it puts that phosphate group on it, it activates it. Okay, now, what is that going to do? Well, that, okay, is subsequently going to then, when it's active, is going to do this. It's going to, and I'm going to again use some abbreviations here, it's going to phosphorylate glycogen phosphorylase. Okay. And that's going to put a phosphate group on there, and that's going to activate that. And you say, what is glycogen phosphorylase again? Glycogen phosphorylase does this. Right? Breaks down glycogen. So what you see here is this. All of this is going on, right? We have glucose going down that then causes glucagon to be released, that then causes pKa to be activated, that then causes fructose 2,6-bisphosphate to be broken down, which activates, right, this step here in gluconeogenesis. And it also phosphorylase kinase, which phosphorylase glycogen phosphorylase, which then activates glycogen breakdown, okay? Right, and then that's going to then allow for the production of glucose 6-phosphate, which then will be made to glucose, and glucose will be shipped out. Okay, so hopefully you can start to see, okay, I'm starting to see some of this. Now, something else that's happening here, okay, is that this, okay, and this is, I'll just have to draw it like this inhibits PP1C. Okay, and you say, PP1C, that's right. Why do I care about that again? Because PP1C will undo pretty much everything that pKa does, okay? Because PP1C is going to dephosphorylate phosphorylase kinase and glycogen phosphorylase and all sorts of things. Okay. Now, something else I didn't mention here is this. I need to draw another set of boxes over here. I'll use another abbreviation, glycogen synthase. Okay, glycogen synthase can also be phosphorylated. Okay, so we put another phosphate on this. It's going to do this. However, and maybe I should have used a different coloring scheme. I think I will. I'll use different colorings. Use red. Okay, it's going to phosphorylate this. It's going to inhibit it. Okay, now what is glycogen synthase going to do? Well, glycogen synthase is going to do this. It's going to make glycogen. All right, so you can see how pKa is like inhibiting pretty much anything that makes glycogen, okay, or he's going to use glycogen, I'm sorry, not glycogen, glucose. pKa is pretty much going to inhibit anything that's going to use glucose. That would include glycolysis because it's removing an activator of glycolysis and it's inhibiting glycogen synthase directly, okay? It will also then activate things that will lead to the production of glucose in the liver so that it then can leave, okay? All right, so lots of arrows here, I know, but hopefully this can start to like gel in your mind. Now, my encouragement to you would be to take this kind of stuff, make tables, make your own diagrams, right? You know, make a comic strip, play taboo with your friends, right? Do these kind of things so that you can start to really assimilate the things that are being presented here, right? I wouldn't ask you to diagram this, but the questions, if you don't have something like this work that you're working from in your mind, the questions on exam 2 will be very challenging for you, okay, to get right, okay? So hopefully you can do that. Now, let me ask, are there any questions on this before I go on? There's more to this, right? I don't know if I have people in the chat that are, let me pull up the chat here. There we go. I do have a question. Okay, doing good so far. Great. So, okay, just let me know if we have questions on this because, you know, you want to get those answered. That's why you're here. Okay, now I will say this along these same lines. There is, before I go any further, there is a table similar to what we have done with our last worksheet, okay, that essentially is, it's actually more extensive, but it's in Canvas in the Exam 2 study modules called Exam 2 Study Aid. I would definitely encourage you to fill out that table, okay? Again, anything that you can do to take what you've learned in your notes and then reconstruct it in your own mind so that you're kind of developing it, you're making it, is going to be helpful in this process, okay? Okay, now let's see if we can't untangle this mess here and do the opposite now. And that would be, what if this happens? You eat a meal. Okay, and you say, oh no, what if I eat a meal? What is that going to do? So that is going to, of course, cause glucose concentrations to go up. In that case, you need to have your body take in the glucose, your liver to take in the glucose, and that means it's going to now, okay, activate this, and activate this. All right, so I'll put little circles around those to indicate that this is when we're eating. Okay, it should also then activate this. Right? Essentially, we're changing red dots for green stars. Of course, you can imagine what it's going to do to the red dots. Okay, I mean, sorry, the green stars is going to make red dots out of them. Right, so it needs to inhibit gluconeogenesis. It needs to inhibit glycogen breakdown. I think that honestly, right, that big picture is something that as I talk to students and press on them, okay, and ask them questions, I think that most people are understanding that bigger picture. It's just all the nitty gritty, icky details that we have to do to get this all done. Okay, now this is where our diagram becomes headache inducing. Some of you say, oh, that's right, it wasn't headache inducing already. So let's see here. All right, let's leave it, let's do this. Okay, glucose concentrations go up and they're going up on the outside of the cell. What that's going to do is this. That's going to cause glucose concentrations inside the cell to go up. Okay, now one of the first things that that's going to do is it's going to cause this. Okay, trying to figure out how to draw this, but it's going to come all the way down here and it's going to bind to this. Okay, and when it binds to that, and I can't really capitulate this, but when it binds to that glycogen phosphorylase, it's going to cause a conformational change in it. That conformational change is going to lead to then these phosphates. There's actually two of them here. I only drew one, but they're going to pop out, and then PP1C, which is bound to glycogen phosphorylase, is no longer going to be inhibited. It's going to be activated. So that inhibition stops, and PP1C will essentially, actually, I'm sorry, let's do it this way. Boom, boom. Glucose binds to this, the phosphates pop out, okay, PP1C cuts them off, and what happens here then is this, right, PP1C causes dephosphorylation of glycogen phosphorylase, inhibits that, okay, and this becomes active. Now, what's it going to do? Well, it is going to do this. Draw another green arrow. It's going to do this. Dephosphorylate that. I guess I should have extended my green arrow a little bit further. I'll do it right now. So it's going to dephosphorylate phosphorylase kinase. That will be now inhibited. So that's not going to be activating, right, glycogen breakdown any longer. And PP1C is going to come all the way up here. Boop. And it's going to cause the dephosphorylation of that, which is going to activate that. Okay, which is then going to go on and make glycogen. Okay, awesome. All right, and that's caused by essentially glucose or glucose 6-phosphate. Both of them will do this, coming into these cells and activating glycogen phosphorylase. Okay, all right. Now, at the same time, as you're saying, doesn't insulin do something? Yeah, it does. And so this is, you know, lots of fun, but we have to have then an insulin receptor in here. So let's draw our insulin receptor right here. We'll make it of a slightly different shape than the glucagon receptor. Apparently tyrosine kinases look just like that, a little bit different, okay? But anyway, this is going to be now, let's draw this, insulin receptor. Right there, okay? And when blood glucose concentrations go up, this is going to lead to, all the way over here, insulin. We'll draw it as a yellow dot, apparently, insulin, and that's going to bind here. Now, dot, 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 right, AKT, right, so tyrosine kinase receptor autophosphorylates, it recruits IRS to the membrane, it phosphorylates it. IRS then activates BI3K, which then makes PIP3 from PIP2. PIP3 leads to PDK, right, activation that then phosphorylates and activates AKT along with mTOR. And then AKT goes on and does stuff, okay? And we're gonna draw some more arrows here. Insulin signaling happens to be fairly complex, okay? But it's gonna do more things. But one of the things that it's gonna do, okay, is it's going to activate this. Okay, PPP, phosphoprotein phosphatase, okay, which is going to remove that phosphate off the kinase domain, and that's going to then put some beams here on the kinase, which is then going to make, boy, this is why you have to really watch out for this, but this is going to then take fructose 6-phosphate and make fructose 2,6 bisphosphate. Right? And then that's going to come here, activating glycolysis. Okay. You want another arrow. You want me to draw another arrow. I'm not going to draw another arrow. Okay, unless there's a question. So that's, when people say, well, how do I tie all these concepts together? Well, this gets pretty close to doing that. So you got to make complicated diagrams. Okay. And I would definitely encourage you to do this on your own time. Okay, all right. Questions on that? Anything regarding that?
So that's all in the liver. Can you go over how that compares to the muscle cell?
We didn't do the muscle cell yet. Some of you
all right, are we going to?
Muscle cell.
Yeah, I'm just making sure.
So.
Okay, thank you.
Okay, it doesn't look like anyone's got questions yet. Okay, so let's do the muscle cell. Okay, now again, I can't emphasize this enough. I've said it a couple times. It's good for me to be it's good for you for me to draw this for you. Okay, it really is. There's I think things that can confirm this with you, but you have to do this yourself, right? And by that I mean the student that's going to get an A on this next exam is going to be the student that's able to, from scratch, draw something like this, okay? Or maybe not draw something, you don't have to necessarily draw it, but you could write it out, right? Some people would rather write, okay, than they would draw, okay? And so they could write all of those things down. Some people will say, I want to put it in a table, and so they'll put it in a table. Other people say, well, I want to teach my friend, or, all these things. I mean, all these kind of things, but you have to find some way where you can get this solidified into your brain. Okay. All right. Now let's go here and I lost some of my membrane when I copied this over, but that's okay. That's the only thing I lost. Okay. And let's do then the skeletal muscle with this. Now, there's quite a bit of complications that I'm just going to remove right off the bat with skeletal muscle. First of all, in skeletal muscle, we don't have this, okay? And I don't think we have this. I think that you can still make oxaloacetate, but that's about it. We definitely have FBPase1. We've talked about that, okay? You have all of glycogen synthesis and breakdown. Okay, so you've got that. Now, we didn't really, let's be honest, we didn't really talk too much about the muscle at rest with regard to glycogen breakdown and synthesis. We did with glycolysis, okay, but we really didn't do it with glycogen breakdown and synthesis, okay? So let's just think about real quickly at rest what we talked about there. And let me use this. At rest, you're right. So your muscle cells, when they're at rest, are going to have quite a bit of ATP just being not used, right? So there will be a fairly high concentration of ATP, and ATP inhibits this. Okay. Now, when we look at this, the converse of that is AMP. AMP activates this, and it inhibits this. right? So if you were to take off right, and do something, okay, take off on a run or whatever, ATP concentrations will go down, right? And AMP concentrations would go up, right? So you just kind of think about it that way, right over here. Exercise, right, leads to ATP goes down, and that then leads to AMP concentrations going up. Okay, and that will allosterically affect PFK1 and FBPase1. Right now, so and we'll just leave it at that. Okay. Now, when we were to start to exercise, okay, so that's kind of all we want to do for rest. We didn't spend a ton of time thinking about rest, though. That's fairly significant right there, what we just talked about. But if we were to go off on a run, okay, or whatever, some level of activity, then lots of things are going to happen. First of all, the thing that's going to happen is AMP concentrations go up. That's going to lead to increased fructose 6-phosphate concentration that's going to feed forward and activate this, which is going to lead to more pyruvate production. Okay, so glycolysis is all of a sudden going to start to really take off when you start to exercise. Okay. And last week in our Q&A, we talked about how it could be this 9-fold increase in glycolysis. Okay, so I don't know if I'll address that right now, but that's certainly going to happen. Okay, right now, the other thing here would be essentially glycogen. Okay, and glycogen breakdown in particular. If we're exercising, glycogen breakdown needs to occur, right? This needs to be positive, and glycogen synthesis needs to be inhibited. Okay. So what's going to do that? Well, there's going to be quite a bit of things, right? So let's look at this. All right, when you exercise, epinephrine is going to be released. Let's use a different color. Not P. It's EP. Okay, epinephrine. Right, and we'll just make epinephrine a yellow dot. Okay, and it's going to bind to a receptor very similar to the glucagon receptor. Okay, it'd be the epinephrine receptor though, right? But essentially what it's going to do is same thing here that we saw in the liver. It's going to lead to the activity or the production of cyclic AMP, and then cyclic AMP is going to activate pKa. Okay, so similar to what we saw up here. Okay, of course I use pink there, but now I'm using yellow apparently. Apparently that's the way we should do this. Okay, and that's going to be now activated, right? Because epinephrine has been released. Now, what is that going to do? Well, it's going to do similar things to what we talked about up in the liver. Okay, it's going to take phosphorylase kinase. And it's going to convert it to phosphorylase kinase active form. Okay, I should draw that with a phosphorylate in it. Okay, now something else to recognize here, and some of you might be saying, doesn't calcium do something? Yeah, it does. When you start to exercise, you start to contract your muscle. What will happen in those cells is that calcium will go up. Well, calcium will be pink. Okay, calcium concentrations will go up in these cells. Calcium can also do this. Activate that. Okay. Well, the way I just drew it is not really great. Let me remove that arrow because that implies that it can activate phosphorylation of PK. That's not really true. It will bind to PK and additionally activate it. So I'll put a little pink plus there just to kind of indicate that. Now, phosphorylase kinase then is going to take GP, okay, and phosphorylate it. Okay. Put a little phosphate here, and voila. Now, that is going to then come and do this, right? Break that down. Okay. At the same time, something else that's going on, right, is glycogen synthase is inhibited by pKa or something else. Okay. Let's just say that. It could be lots of different things. Put a phosphate here, inhibits this. pKa will inhibit that, okay? AMP kinase will do this. Okay, phosphorylate that. Something called CAM kinase, which is activated by calcium, will do this. So you have CAM kinase. We'll do this. CAM K2 will do this. And calcium will activate that. And then this will do this. Okay, so there's lots of ways to essentially phosphorylate glycogen synthase. Okay, we could have done that up here too. We could have put several ways. One of the ones that I'm really ignoring and that I probably shouldn't is GSK3, glycogen synthase kinase will do that too. So, okay. So glycogen synthase is fairly complicated in terms of its regulation. Now, again, what is it going to do? Well, it goes and does this, right? Makes glycogen. So that needs to be inhibited. Okay. Now, the last thing that we probably want to talk about here would be PP1C, because PP1C can go in and, do all that, dephosphorylation stuff, right? Well, how is that going to be inhibited? Well, that is going to do this, or how that's going to happen is this, okay? Running out of good colors. Pink. We're going to have to make this pink. Okay, we have that thing GM that binds to PP1C and a whole bunch of other things. Well, pKa is going to phosphorylate it. Okay. And that phosphorylation is going to cause PP1C to be released. PP1C release. Okay. Now, at the same time, pKa is going to phosphorylate inhibitor protein. I'll put I in pro, inhibitor protein, that then is going to inhibit PP1C. Okay, and that will then prevent PP1C from undoing everything that pKa or AMP kinase or CAM kinase is doing. So, okay. Okay, so that's, I think, a pretty good description of much of what we did in lecture 6, not all of it, but much of it. Okay, don't forget that we talked about how insulin can be released from the pancreas. We went through that entire pathway. We talked about insulin and glucagon signaling, so don't forget to look at those. Okay, and of course we did, all of this, right? We did also cardiac tissue with FBPase2 and PFK2. Okay, this is skeletal muscle. I didn't do the cardiac muscle. Okay, but we did that as well. Okay, so let me ask you this: questions? Are there questions? Are there other questions besides what we just went through? I just realized that took all of 45 minutes to do that. So. No? Okay, yeah, so Rachel's got a question. For problem set 3, there are 3 irreversible steps, correct? Absolutely. That's correct. In the citric acid cycle. Yep.
And
other questions?
So for lectures 7 and 8, we're talking about pyruvate dehydrogenase.
Yeah, so
then,
Marissa just asked, is GSK3 an inhibitor of glycogen synthase? Yes. So if we were to come back here, we could put GSK3 on this list as well. We could put it up here as well. And quite honestly, we could have put some of the ones down there up here, right? So I could have put like AMP kinase, although it's not as important, okay, or CAM kinase up here as well, okay. But yes, GSK3 is definitely that. Yeah, of course. Other questions? Remember today, okay, to get credit for being here, you need to put something in the chat, okay, even if it's see you later. That's fine. So, is there any questions on, like, for instance, lecture 7 or lecture 8, which we're kind of in the throes of that, okay? Yeah, Cole. So Cole asked, lecture 7 and 8 are all about how pyruvate is consumed. That's, yes, that's true. So in lecture 7, we see how pyruvate is converted to acetyl-CoA, okay, by pyruvate dehydrogenase complex. And then that acetyl-CoA then feeds into the citric acid cycle, which then, right, essentially converts those carbons to carbon dioxide. The pyruvate dehydrogenase complex reaction also is what we call oxidative decarboxylation reaction that then removes a carbon from pyruvate making acetyl-CoA. Okay, so yes, it is about that. Now, I will say this, Cole, in particular with the citric acid cycle, that is like the hub of metabolism. So when we think about the citric acid cycle, that's going to come up time and again. We're going to see how the citric acid cycle is very important in lipid metabolism, is very important in amino acid metabolism. If we were to go through nucleotide metabolism, it would be very important in that as well. So at the moment, we have connected it to pyruvate, okay, consumption, but we're going to connect it to lots of other things as well, okay? And that's why we talk about those anaporotic and cataporotic reactions in the classes, or in lecture 8. So, okay. Right. Hunter's got this question. So for enzymes with multiple phosphorylation sites activated by the same similar enzymes, do the sites typically have similar effect, or do different changes, or do different changes to the molecule? So if we're generalizing, the answer would be yes. So sometimes the additional phosphorylations can just be simply that. They can just be more of the same. Many times, though, it's usually different. Okay, it usually causes a conformational change that then maybe sometimes even reveals an additional phosphorylation site that then can lead to greater and greater inhibition, right? So if we go back to this idea that enzymes are more like dimmer switches, right? Phosphorylation can, get that dimmer switch can move that. It can be moved up or down based off of, the types of phosphorylation that are happening. So that is a very context-dependent answer, quite honestly, Hunter. It just really depends on what you're looking at. So I would say generally, though, it's more of a cooperative effect, meaning that the more you get, the greater the effect. So. Okay. Marissa, is quiz 8 up through lecture 8 information? Like once we finish both parts of 8B, should we be set to take it? I would have, that's a great question, Marissa, that I of course, I wrote it and prepared for it, and I've forgotten what exactly questions are on it. Let me look real quick. I have to log into Canvas, but let me look at that real quick, okay? For us.
I asked more because we
give you a better answer.
Lecture 9 in almost an entirety before the quiz is due.
been a while since I looked at quiz 8. It was a couple weeks ago, so I just can't remember what's on it. Yeah, so you'll wanna go ahead and finish out most of lecture 8 before you take it. So okay. Yep. Other questions? Anything else? Okay, great, Carter. Yeah, so it's that time. It's 3:53. So I have to start another Q&A session with my other class at 4. So if you really don't have anything else, then you are free to take off. I hope that this was helpful for you, and that you got some clarity on some of this stuff. So.